Okay, so let me briefly introduce Kirill. So Kirill is he received an MD at St. Petersburg State University in Russia and started a PhD in University of Iowa. And I started doing a postdoc with Ralph Adolphs, who moved into Caltech around the time Kirill probably started postdoc over there, I think. And since then, I have known him, on and off. Initially didn't talk much, but because he was mainly working on the auditory paradigm, and I was working on the visual paradigm. But now, eventually, our interests kind of crossed quite a bit. Now he's also looking at intracranial recording difference between wake versus anesthetized or sleep. And but his main interest is, I believe, still auditory processing and how our sound experience arises from the brain. And so he's also been quite responsible for many of the experiments going on in Iowa. So if you have any logistic kind of questions, he will be able to answer pretty much everything. Okay, thank you, Kirill, for agreeing with the talk. Please go ahead.
Yeah, thank you for the introduction now. Thank you for this invitation. It's an honor to present and very unusual experience. So I will talk over the next 40 or so minutes about how does the brain respond to auditory stimuli and what happens to resting state connectivity in the brain as we examine different awareness conditions and different task conditions. And we'll use intracranial electrophysiology in humans to address some of those questions. So,
wait, I forgot to ask you, sorry. Should we encourage people to interrupt and ask questions through chat during the talk, or just raise a hand and so on?
Where is the chat? I think you can just interrupt me. I mean, it's not like we have 100 people here. If it's okay with you, I don't mind being interrupted.
All right. Yeah, you can just speak, or you can also, raise question in the chat. And
I don't even know where the chat here is, because one of my windows is my own presentation. So yeah, just basically, yell. Anyway, so just as a bit of a background, one of the major topics in modern neuroscience, of course, is understanding what are the neural signatures of sensory processing that define different awareness states, awake, unconscious, and so forth. And conditions that are clinically relevant to this topic of investigation include sedation under general anesthesia, sleep, and disorders of consciousness, such as delirium, for example. One of the fundamental functions of consciousness is the interplay between the predictions, the sort of top-down predictions, and the bottom-up sensory observations, the comparisons that take place in the brain. And of course, that forms the basis of the so-called predictive coding framework that has been, in the recent years, has been very strongly kind of adopted, utilized in auditory neuroscience. And I'm an auditory neuroscientist by my trade. And of course, disruption of the predictive coding mechanisms in the auditory realm specifically may be considered a signature of unconsciousness under general anesthesia, right? And in addition to that, based on the available evidence, we can argue that changes in connectivity in the brain also likely contribute to loss of consciousness. In the past, there's been a lot of non-invasive studies using scalp EEG, functional MRI, PET, and so forth that have shown that in the auditory domain, there is a reduction in activity in non-primary as opposed to primary auditory cortex under specifically propofol sedation. So there are several studies that examine that. Now, I work in a neurosurgery department with a team of neurosurgeons and scientists, and I have the unique opportunity to address some of these questions by recording directly from the brain in epilepsy patients who undergo implantation of electrodes to refine the seizure focus prior to surgical treatment of their seizure disorder. So in this method, intracranial electroencephalography, or iEEG, offers really a unique opportunity to study cortical activity with great spatiotemporal resolution. So why auditory and why, auditory and consciousness? Number one, oftentimes about 70%, I think, of focal seizure disorders have a seizure focus somewhere in the temporal lobe. And that, of course, warrants the electrode coverage of the temporal lobe, and that, of course, is where auditory cortex is in humans. And also, I would argue that if we want to study consciousness and like loss of consciousness under general anesthesia or during sleep, the auditory modality is, for example, more as a kind of a sensory modality, as a tool to probe sensory processing. It makes a lot more sense than, let's say, visual, because, well, first thing that happens when people start drifting away is obviously they close their eyes, don't close their ears. So with this approach, we have a unique opportunity to access primary auditory cortex in the posterior medial portion of Heschel's gyrus, right, kind of deep there in the sylvian fissure, as well as adjacent non-primary auditory areas, including anterolateral portion of Heschel's gyrus, planum temporale, planum polare, the lateral surface of the superior temporal gyrus, adjacent temporoparietal auditory-related cortex, and prefrontal areas, and everything in the kitchen sink. This method has been utilized in our lab in the past to demonstrate that there is a regional heterogeneity in the effects of general anesthesia on responses to simple stimuli, such as click trains. So some 20 years ago, our department chair and the overall director of the research program here, Dr. Matthew Howard, has demonstrated that when we present click train stimuli in the operating room to these patients prior to removal of the electrodes, he found that in the primary auditory area, responses, the short latency responses to these stimuli are relatively unchanged, whereas responses on the lateral surface of the superior temporal gyrus, which is kind of a secondary, tertiary auditory field, are greatly affected by general anesthesia. More recently, my colleagues and I did this study where we, again, we use click trains, and we looked at the so-called frequency following response, basically the ability of the brain to track the regularity of the click train. And we found that in the most medial portion of Heschel's gyrus, there is actually an increase in the strength of this frequency following response, which we attributed to basically kind of an unmasking effect, that the incoming regular phase-locked activity from the thalamus reaches the earliest auditory areas, and it's not really masked or confounded by any other kind of concomitant ongoing processing.
Kirill, just a quick question.
Sure.
So, as for the left study, the baseline to third epoch, it's the progression of the depth of anesthesia, or it's just
Yep, that's basically, that's awake, that's right before, and these are just 3 time points after bolus delivery. I think they used thiopental as opposed to propofol.
Okay, and then the other side is that is this in a sense sort of the predictive coding like paradigm?
No. This is a very boring paradigm where the subject just hears the same click train over and over and over and over again.
And the increase means that the repetition increases the amplitude of the response.
So it's basically the strength of the phase lock response. It's how accurately, basically, if you have a 50 hertz click train, right, you can measure. So a 50 hertz click train will have a 20 millisecond regularity. There'll be a click every 20 milliseconds, right? And responses in the cortex, in some parts of the cortex, will kind of have that component that kind of has this like 20 hertz periodicity, and you can measure the strength of that component at 50 hertz, right?
I see.
So it's kind of a very low-level isomorphic representation of the stimulus. But anyway, that's the past history. That's boring. So more recently, we utilized a more advanced paradigm, a more complex paradigm to probe these predictive coding mechanisms, which, as I introduced in the first slide, we feel are central to kind of conscious sensory processing. So we use this so-called local global deviant paradigm that offers the advantage of engaging predictive coding on multiple timescales. So this is, I apologize, this is a little bit, if some of you are not familiar with it, this is a little bit complicated, but it's critical for everything else that we're going to talk about later, right? So imagine the elementary unit here is a stimulus that includes 5 vowels, and it can be either all 5 vowels are the same. Whoop. Did you hear that?
Yeah.
Or the 5th vowel may be different from the first 4, right? So whether the 5th vowel is the same or different as the first 4, that constitutes so-called local deviance or short-term novelty, right? So it's a pretty, you can think of it as like stimulus-specific adaptation. Like you present a 5th stimulus that's different from the first 4, you recruit a fresh batch of neurons that are not adapted by these preceding 4 stimuli. Now, when you start playing these stimuli in a sequence, right, you present this one, this one, this one again, and then you diluted this one, right? So how frequently or infrequently this one or this stimulus is presented within a whole sequence, within the whole experiment, that constitutes long-term novelty or global deviance, right? So it kind of has these two levels built in. Now, post hoc, like after, and this is balanced by the experiment is designed such that these different dimensions of the stimuli are balanced. So then we can take like a bunch of trials that correspond to local standards and compare the responses to them to local deviance. So they're going to be matched along the dimension of global deviance and the vowel identity. And we can do the same thing for global deviance. We can take all the global standard stimuli that are presented frequently and compare responses to them to global deviant, that is infrequently presented stimuli. We can basically reveal any difference at that 5th vowel can be interpreted as a so-called local or global deviance effect. Does that make sense?
So just a question on global deviance, sorry.
Sure.
So with global deviance, are we talking about the time scale of the whole experiment? So you get your patient in, you sit them down, and global deviance is measured in a scale of 10 minutes, half an hour?
Yes.
Or, yeah.
Yes, that is exactly. This is one of the dirty secrets of this paradigm, is that unfortunately you can't really like slice and dice it and take you like 3 trials here and then 3 trials there. In order to make sense of these data, you have like, I have this, because you have to alternate, you have to basically establish this as a global standard. Then you have to play it, and every once in a while play the deviant, right? Then you have to switch it, and then this becomes your global standard, and this is a global deviant, right? And then the order of these 4 sequences is randomized, but you have to get all 4 to make sense of the data to basically make sure that whatever you are subtracting from here is actually balanced along the other 2 dimensions, right? So you can only analyze the data from the entire, in this case, 11-minute block.
And Kirill, did you instruct subjects about this?
Excellent point. So the task we used in this experiment initially was a target detection task. They were supposed to press a button whenever they heard the global deviant stimulus.
Global deviant, okay.
Global deviant, yep. So basically, before each of these 4 like 2.5-minute sequences begins, there is a verbal instruction that says, now press the button every time you hear this sound, and we give them a preview of the global deviant. And then when we switch, we say, now press the button every time you hear this sound. So that was
that's okay. Then naturally means that you train the subject with this thing under the awake situation.
Yep.
And then test it in the anesthesia.
Well, no, we train them. We train them beforehand. It's a pretty easy task to teach them, right? So we do one 11-minute long experiment in the awake state, right? They press the button, perform the task. Then we start propofol. Actually, if like there are no questions regarding the structure of the stimuli, we can just proceed to the actual experiment structure, right? So here's what we do in the operating room the day when the subjects go in the OR to have their electrodes removed. We can we design this slow infusion paradigm where First, we have a baseline, about 15 minutes, where we run this experiment in the awake state. Then we start propofol at 50 microgram per kilogram per minute, and then we gradually increase the propofol level. And as you can see, that is associated with a gradual decrease in BIS, which is one of the metrics that can allow for assessment of depth of anesthesia. In addition to that, so while this happens, we present 3 or 4 11-minute long LGD experiment blocks, right? So this is going to be the awake one, where the patients are pressing the button every time they hear the global deviant. This is where usually they start failing because they get drowsy, and towards the end they usually just flat out unresponsive, right? So then what we can do is we intersperse these auditory experimental blocks with resting state blocks, where it's just like no task. So these data are about 5 minutes long. We collect them for the purpose of examining resting state connectivity. So after that,
I have a quick question. Sorry to interrupt.
Nope,
but
no.
This means that in the context of this anesthesia experiment, the global deviant is set at the beginning and never changes. Because
no, it does. It does change every 2.5 minutes. Remember, that's why we have 4 sequences here, right? So
in the training, you teach them the concept of global deviance, but then which stimuli exactly is a global deviant is flexible throughout the task, right?
Right. So the global deviant is defined. So the 11-minute long block is it consists of 4 quarters, right? And at the beginning of every quarter, we define the target stimulus, the global deviant, to the listener, right? It's not like a separate training session. It's all part of this experiment. Make sense?
Thanks. Yeah.
Right. So also in between blocks, we do the observer's assessment of awareness and sedation, which is kind of a scale 1 to 5 clinical tool to assess whether they are fully awake or completely unresponsive. Then we use the OAS as our main OAS, and also lack of behavioral responses to the target stimulus as our criteria to define each of these blocks into 3 brain states. Either awake, that's easy, right? That's going to be everything pre-propofol. And then sedated, that's past beginning of propofol infusion, but while they're still responsive, and we define it as, this is going to be greater than 2. So that's 3 and above, and this is 2 or less, right? And then we basically pick our 3 blocks that unambiguously define our awake, sedated, and unresponsive condition, and we compare neural activity between these 3 blocks. So we do that. This experimental paradigm allows us to accomplish several research objectives. Well, first we can look at changes in responses to these vowel stimuli, right? Like the first 4, there is no surprise, there is no novelty there, right? So we can just look at what happens to responses to these during induction of general anesthesia. We can also refine the localization of responses to auditory novelty by focusing on this temporal window, right? In the awake state, we can see how these vowel responses and local global deviant effects are sensitive to general anesthesia and in addition to task. And then we can examine changes using these data, we can examine changes in resting state connectivity that occur under general anesthesia and sleep. So that's basically the 4 parts of this presentation. First, I'll show you a little bit what happens to auditory responses to the first 4 stimuli. So that's in each trial, the first 4 vowels before any kind of novelty sets in, right? We report from these multiple regions. We'll look at evoked potential waveforms and high gamma power envelopes, and examine the significant, basically the significant deviations using cluster-based analysis. We find that these auditory stimuli presented in an active task, they elicit robust evoked potentials and high gamma responses in the auditory cortex that are preserved across awareness states. So you can see even in unresponsive, there is still plenty of activity. In contrast, responses in auditory-related adjacent cortex, for example, middle temporal gyrus, are weaker to begin with, and they get even smaller in sedation and loss of consciousness. Finally, we also find that the early auditory cortex right here features so-called envelope following responses, right? So there are 4 vowels here, and you see 4 peaks in the response, which is kind of almost reminiscent of like tracking of the syllabic structure of speech that's been described for auditory activity. When we combine data from multiple subjects and project sites separated by region of interest that exhibit those significant responses to vowel stimuli, we find that, well, there's actually, the activation is quite widespread. The responses are broadly distributed. High gamma responses, in contrast, are quite a bit more restricted. And in summary, just sedation and loss of consciousness are associated with decreases in the number of responsive sites in the prefrontal cortex and auditory-related cortex to a greater degree than it does in the earlier auditory areas. Temporal, if we examine temporal profile, and that's basically counting the sites from all subjects that at any given point in time relative to the stimulus onset on a trial-by-trial basis, how many of them exhibit significant responses, we find that these changes from awake to sedated to unresponsive mainly occur at the expense of these higher order auditory-related and prefrontal areas. And these are just some boring, bar graphs to show you the changes in percentage. We find that using chi-square tests, so that's like from all of our subjects that we examined how many sites exhibit the significant response. We find that there is a progressive decrease in percentage of responsive sites along this cortical hierarchy from early to secondary to auditory-related cortex. Sedation is associated with decreases in percentage of sites with significant evoked potentials and high gamma, and loss of consciousness is characterized by further decrease in percentage of responsive sites in auditory cortex. As an additional analysis, we also looked at the magnitude of response and the findings here in general, as well as latency. The findings here in general are consistent with what we see when we just look at like the prevalence of responses, right? So it's in a nutshell that this can be summarized as decrease in the percentage of responsive sites, decrease in response amplitude, increase in response latency. Also, curiously, when we examine this envelope following response that we saw in primary auditory cortex, we find that it actually does significantly decrease upon loss of consciousness if we compare the sedated and unresponsive states. So that's the kind of the summary of that part, maybe like the least interesting part before we even talk about any predictive coding. We find that in the awake state, there are evoked potentials throughout brain areas. High gamma is more focused in the auditory cortex, and there are prominent envelope following responses in auditory core. Sedation is associated with a decrease in the number of responsive sites and response magnitude, increase in latency. The biggest changes occur in non-core auditory and auditory-related cortex, so that potentially may hold promise as some sort of a biomarker for loss of consciousness. And activation within auditory cortex by these vowel sequences presented in an active task is actually a lot greater than what we had previously found using just simple click-trained stimuli presented in the passive paradigm. So now moving on to auditory novelty, let's first take a look at what happens to these responses in the awake state, right? So now we are concerning ourselves with this, the final epoch, the fifth vowel, which may or may not constitute local or global deviance, right? And we find that auditory novelty elicits these evoked potentials and changes in gamma responses by comparing responses to standard and deviant stimuli. We see these effects. It just goes beyond offset responses to the final vowel.
Sorry, Kirill.
Go ahead.
Are you able to just quickly give a layman's definition of evoked of AEP and high gamma, the difference?
Oh, sure. I'm so sorry. So an evoked response, basically, you take your raw voltage, right, that goes positive to negative, that you record from a recording electrode, right? Then you take that, let's say, 11-minute long recording. You find your timestamps that basically the onset of each standard stimulus, right? And then you do time domain averaging of those chunks of your LFP recording with respect to that time zero, right? With high gamma, actually here, let me just show you a picture may say maybe more informative here than just me waving my hands and doing an introduction here. Just yeah. So this is how you get the evoked potential. You do trial 1, trial 2, you average them, get the evoked potential. Make sense? For high gamma, it's a little bit trickier, right? Because high frequency activity, it is time locked, meaning that it does basically change upon, it's related to timing of the stimulus, but it's not phase locked. So if it's random phase, by doing time domain averaging, you average it out. So to get at it, what you want to do is you want to rectify your signal, preferably in the time frequency plane. So you basically compute spectrograms. Then you normalize these spectrograms to whatever power at each frequency there was prior to stimulus onset. And then you average your spectrograms. You get your average normalized spectrogram. And from there, you can take a slice that corresponds to high gamma, which is 70 to 150, right? And that becomes your high gamma envelope. Does that make sense? So this basically, you can think of AAP. AAP is basically summed postsynaptic potentials, and high gamma can be considered the proxy of like spikes.
Yeah, that makes sense.
Yep. All right, and so these are key. These are two of our key metrics here. So we find that both local and global deviant effects are widely distributed across multiple cortical regions. They're not limited to auditory cortex. We find that they exhibit a spatial overlap when we kind of plot the plot data from multiple subjects and look at where do those effects take place. We see that local deviant effects in auditory cortex are usually associated with global deviance effects, and sites that only exhibit global deviance are rare. Sites that are exclusively responsive to global deviance are prevalent in auditory-related and prefrontal cortex. Now, superior temporal plane right here largely exhibit only high gamma local deviant, but not global deviant effects. And we don't really see.
Yeah, Kirill, so here this is all awake, right?
Yep, this is all awake.
And all analyzed with correct responses?
This is regardless of their task performance. This is we take all the local deviant trials, right? So for local deviance, there is no such thing as correct or incorrect response because they're not really responding to local deviance.
But you can have a false alarm as well, right?
Yeah, there's plenty of those, but we don't really, we only use the behavioral responses as a way of kind of additional insight into whether they are awake or not.
Yeah, but why I'm saying that is that it may be interesting to look at the global deviant, which was not detected.
Oh, right. Yeah. Well, again, getting back to the previous comment and getting back to the complexity of this paradigm, it's really, you really can't pick and choose your trials because the trials have to be balanced, right? Because of this. It's a little, I know that the group that designed this task, they do, like in one or two of their papers, they do have ways of kind of looking at this at a finer grain, but I kind of tried to, but I think this really works only when you, like, basically for every local deviant, you have to have a local, like, it's complicated to kind of do a fine grain analysis.
I see. Then the second quick question is that do you have an awake but no response situation?
Yes, and that will be part 3B of this presentation. Right. We're getting there. Okay. Anyway, so quickly, an interesting thing we found was when we looked at the onset latencies of the local and global deviance effects, and we found that for local deviance, it's pretty much bottom up, right? So as you go along the auditory hierarchy, you get longer onset latencies, so that's consistent with feed-forward processing. In contrast, global deviance has a more complex time course where the shortest latencies are in the superior temporal gyrus and adjacent auditory-related cortex, and then it kind of branches out, it goes further to prefrontal cortex and also down to early auditory cortex. I thought that was pretty neat. I already talked about spatiotemporal patterns. I'm sorry, I'll start kind of skipping some of these less interesting slides for the sake of brevity. Just to summarize this, that's still awake business, right? We find that novelty detection engages multiple cortical regions, and local deviance effects are associated with mostly feed-forward information flow. Global deviant effects likely originate in STG and then go forward and back to early auditory areas. Now, we talk about changes in this business that we talked about so far, of course, levels of awareness and task conditions. So, when we continue this experiment, right, after we start them on propofol, and we can compare the sedated and unresponsive conditions, we find that it is local, but not global deviance effects that are preserved in early auditory cortex upon induction of general anesthesia. And in general, we find that local deviance effects in auditory cortex are fairly robust and resistant to propofol anesthesia. So these are data from 6 subjects, and each bar is a block. I label them for your convenience as awake, sedated, or unresponsive. And you can see that the number of sites in each of these regions of interest, they change, but they mostly change at the expense of these higher order greens and blues. That's the auditory-related and prefrontal cortex areas. But in early auditory cortex, they're fairly robust. Now, global deviance exhibits a very different pattern. We see that we lose it early, and loss of global deviance responses actually occurs before loss of consciousness. So we find that local and global deviance are differentially sensitive to propofol anesthesia. You see that, you know, the biggest change here occurs between, obviously, like the clinically relevant divide is this one, right, between sedated but still conscious and unresponsive, as opposed to this one. So we see that here, indeed, the big difference occurs in the frontal lobe and in the kind of temporal cortex, whereas the biggest change for global deviance occurs way before. So we find that.
Sorry, I had just a quick question regarding the cessation of global responses. How do they relate with the moment where you start having infrequent responses or a decent amount of misses?
Excellent point, and we actually followed that up and wrote a paper about it, and I'm about to show you that.
Okay,
right,
because
so,
I'm aware of a paper on the same paradigm in sleep using MEG, and they have this disappearance of the global event, pretty much when people stop responding.
Right. Yep. So we see. From the anesthesia data, we see that general anesthesia propofol has different effects on local and global deviance, right? The local deviant effects are preserved within auditory cortex and suppressed elsewhere under propofol. Global deviant effects are suppressed prior to loss of consciousness. So we can make this conclusion that the presence of global deviant effects is indeed indicative of being awake, but their absence is not really predictive of loss of consciousness. Well, could it be that what we found with global deviance is really reflects more task engagement than consciousness per se, right? So we examined that by doing another set of experiments, not in the operating room, but in the clinical research unit while the patients are still, you know, awake. And we ran this paradigm twice with them. Once when they were listening to these sounds without any task, and they were actually they were asked to watch TV and pay attention to the TV program. And then we did the same experiment in each of those patients. We did the same experiments when they actually doing the same thing as what they were doing in the operating room, right? Doing a target detection task. And what we found was responses to vowels, so prior to the onset of novelty, are very similar. So here, I guess the most informative here will be this diagonal, right? So I'm plotting for each of the regions of interest. I'm comparing the percent, how many sites have a significant response in the active states on the y-axis versus x-axis, the passive, right? So if it's on the diagonal, no difference. If it's above the diagonal, then there is a bias that is strong in the active condition. That makes sense? So while responses are pretty robust, and of course the strongest in the early auditory areas, as one would expect, local deviance effects are slightly more prominent in the active condition, but really not much to write home about. Now, if we look at global deviant effects, that's where things get really interesting, and we find that indeed they are much more prevalent in the active condition, as opposed to awake but passive. So this support, I hope this, Thomas, I hope this answered your question, and I would argue that this supports our hypothesis that global deviant effects really are more related to task engagement. What's more is in this paper, this is like, right, our anesthesia paper was 2017, that's old news. This is the newest stuff. This is currently in press hearing research. We started looking at alpha suppression. So these active paradigms, they often associated not only an increase in high gamma power, but also a concomitant decrease in the alpha power associated with stimulus presentation. And we found that while alpha suppression that is associated with local deviance is fairly comparable between the passive and the active condition, for global deviance, it's almost exclusively present when subjects are engaged in a task. So that supports the role of alpha suppression in functional inhibition mechanisms and communication of sensory predictions. It's basically you kind of shut down all the relevant stuff and focus on the task, and that's what alpha suppression is. So we found that from this part, we found that vowels and local deviance elicit robust evoke potentials everywhere, high gamma responses to stimuli, and the local deviants are mostly localized to early auditory areas, and they really have a comparable prevalence and special extent between the two conditions. Global deviants are greatly, greatly enhanced when the subject actually is performing the task. And again, that supports the hypothesis that global deviance is strongly driven by task as opposed to consciousness per se. Finally, this is one of my favorite children. You know, I consider all my papers as children. This is definitely one of my favorite kids, where we looked at resting state connectivity during anesthesia and sleep. So remember when we were doing the anesthesia experiment, we had these resting state blocks, right? So also in the same subjects, we did this study in five patients. In the same subjects, we recorded their intracranial activity during overnight sleep, while they also had scalp EEG leads placed, and that allowed our sleep research colleagues to do sleep stage scoring for us. So we could label their sleep epochs according to like N1 and 2, wake, REM, and whatnot. So intracranial method's pretty much the same, record from as many areas as we can. Then what we do is we look at functional connectivity using weighted phase-lag index, which is a non-directed measure of phase synchronization that accounts for and kind of discards synchronization near zero phase lag, because that usually that's more likely to be volume conduction, or like two electrodes see a signal from a common source, rather than evidence for an actual functional connectivity between the two. So my friend and colleague Matt Banks, who led this study, he computed these connectivity matrices in the alpha frequency band for all pairs of recording sites for each subject. Here is an example from one. So this is in the wake state, right? So you go contact by contact, there is like 200 of them. Then you group them by region of interest, you get a more coarse matrix. And then you take this and kind of turn this into a little bagel, and you connect, you build a chord connectivity plot that basically tells you the strength of relative strength of connections between different regions in this state, right? So that's what it looks like in this subject in the wake state. This is what it looks like for 5 subjects across different states. So what we found was transitions into states of reduced consciousness, be that N2 sleep in the sleep study or unresponsive in the anesthesia study, are both characterized by reorganization of connectivity, and this reorganization is remarkably similar between the two states of reduced consciousness. Specifically, what we find is there is a transition from mainly temporally kind of connectivity patterns that are driven by temporal cortex, as shown here, all these greens and yellows. And then as the brain passes this boundary from N1 to N2, or from sedated yet responsive to unresponsive state, we see this shift from temporal to predominantly frontal connectivity patterns. Moreover, when we used a linear classifier and we trained it on sleep data of the wake epochs from the sleep experiment and the M2 data from the sleep experiment, simply because we had a lot more data from obviously from the overnight sleep experiment than from anesthesia. There was and basically the logistic prediction of data segments basically tells you whether a given data segment looks more like a sleep or awake based on this training, right? We find that it actually does a really good job both for sleep and for anesthesia data.
Oh, wait, Kirill?
Yep, go ahead.
About the averaging method, so here you average at the level of the grouped ROI matrix and then get one of these, graph pattern, or did you get the graph pattern per subject? And then at the level of the graph pattern, did you average? Did it make sense? Make difference?
Right. The order of averaging, that's an excellent question. I'll be honest with you, I do not remember in which order did we do that. Matt Banks would know that. He did that. I don't remember.
Maybe it's just a visualization, so it may not make that difference.
Well,
all right,
also now, speaking of visualization, I should make a point that these are thresholded. I'm only showing the 10% strongest connections here, because otherwise it would be like a spider web made by a drunk spider. It would be like impossible to make sense of.
Yeah.
Okay. So we found that reduced arousal really is associated with this anterior bias in connectivity. And we also looked at, there are these, some previous studies argued that loss of consciousness is associated with the decrease, with the change from between long range and short range connections, that connections become more local. Well, in fact, we kind of found the contrary. We found that there is a very modest increase in long range connectivity. But this finding the posterior to anterior bias in the connection patterns, that was very robust. And much to our delight, we found, and now I'm sure you're well familiar with this study, right? That, that kind of was very much reminiscent of this whole concept of neural correlates of consciousness, that's basically your temporal occipital cortex, right? And that's what's active when you are conscious, and that's what can basically becomes sidelined when you're not. So we thought that was a really neat kind of consistency with that theory.
Excellent.
This temporal to frontal shift of alpha band connectivity, we argue that it's like this transition boundary between states of maintained and reduced conscious experience. And what does this mean? It may reflect impaired sensory processing. And we found that this reorganization is consistent between sleep and anesthesia, meaning that they kind of share common mechanisms. And what's promising, like from the clinical angle, is that it may hold promise as a biomarker of arousal state that can be used. So just to sum it all up, go ahead.
Sorry to interrupt again, but regarding this alpha, this shift to frontal areas for alpha connectivity, it's very interesting that you have the same in anesthesia and sleep. But I was wondering, the type of anesthesia, I'm not a specialist of anesthesia, but is it the type that's generating these strong alpha oscillations in the front? That
is an excellent question. There are two projects underway. Had I finished them, I would have showed you the data right away. But yes, basically, you can basically say, well, is this just the effect of propofol? Right? Is this a drug effect? I don't think it is, because we see this similar patterns in sleep, and there is no propofol there.
Yeah, but for the sleep, I'm wondering if you label as alpha oscillation something that could be closer to spindle.
Oh, like you're thinking like spindles?
Yeah. Because in frontal areas, they overlap with the alpha rhythm.
Yeah. There was, I think we did have that concern, and I don't remember how exactly we addressed it. My fault, but I think it had something to do with, we examined power, right? Because if you have alpha spindles, you would also see a dramatic change in power.
Yeah.
Right? And changes in connectivity, which is the way this is computed, it's kind of, it really is unrelated to power changes per se, right? It's synchrony, not power, right? Those are much more robust than any changes in the just the spectral profiles.
Okay. Okay.
But that's an excellent point. But also regarding propofol and sleep, of course, we have, and that's something I have in the overall conclusions that is coming up, right? So just to sum everything up, and I'm sorry, I apologize. I get too excited when I talk about my work. I just want to have this urge to tell you everything, and I run over time. But the take-home messages from this are, we find that sensitivity of auditory cortical activity to general anesthetics increases along the cortical processing hierarchy, perhaps not surprising. We find when we study auditory novelty in predictive coding that presence of global deviance is indicative of being awake, but their absence is not really a biomarker of loss of consciousness. As far as potential alternative biomarkers of loss of consciousness, maybe based on all these 4 or 5 studies, those could be a decrease in responses to sound and loss of local deviant effects in higher order cortical areas, what we call auditory-related and prefrontal cortex. Second is that curious finding that the envelope following becomes limp in the early auditory cortex upon transition to unresponsive state, but not before that. And also this robust temporal to frontal shift in alpha bad resting state connectivity. Now, all these findings, of course, there is a long road towards any kind of clinical applications, but we feel that, my colleagues and I, we feel that they can serve as a foundation for further exciting studies that would examine changes in sensory processing in the cortex and connectivity. Specifically, of course, an outstanding question, well, will this generalize to other agents? For example, Precedex, or something like sevoflurane, the inhalation agents. Natural sleep, we are expanding our research program to beyond, on the one hand, we're looking at novelty responses during daytime naps, as well as we're comparing connectivity patterns during daytime naps and overnight sleep. And of course, another very important clinically relevant topic is disorders of consciousness, such as delirium. So we've been working with our patients and studying their kind of monitoring them for post-operative or post-ictal delirium and examining patterns of cortical activity under these states. So with that, I would like to thank you all for your attention and your excellent questions and this opportunity. And I need to thank my esteemed colleagues without whom this would not be possible. My friend and colleague Matt Banks, who's my main collaborator and driving force in this research program. He's at Wisconsin-Madison. Dr. Arian Rohn, who is an incredibly helpful and smart colleague here at Iowa. Mitch Steinschneider, my old buddy at Albert Einstein, who is like started as a fatherly figure to me, but now we just write papers with him on Skype and on Zoom 5 days a week. And Dr. Howard, of course, who is the director of this program, and funding generously provided by National Institutes of Health. Thank you very much.
Thank you. It was very exciting.
Oh, this is the kind of applause I get.
And you also get upload icons from.
Oh, I see. I didn't notice those. Okay, yeah. Thank you.